Thank you, everybody. How do I follow that? I guess I have to energize you and get you all awake now. So, my name is Daniel Harvey. I'm with the Angelman Syndrome Foundation. Been on the board for a long time and have been involved in the science of AS for decades. It's an honor for me to introduce the first speaker in our next session, Emil Kakkis from Ultragenyx. Emil is not just the president and CEO of Ultragenyx, he's the founder of the organization. They were founded in 2010 with a focus on the development of treatments for rare and ultra-rare genetic disorders, so an area that we are very excited about here. They've had quite a bit of success. They have 4 approved therapies, 4 or 5 different rare diseases, and they have multiple late-stage programs, including an ASO-based treatment for AS that is in the clinic that we have heard about some this morning, and we're going to hear about more this afternoon. Emil has a lengthy list of awards, and if I read them all to you, he wouldn't have any time to speak. So I won't mention those, but I did notice as I was reviewing his bio that he was a graduate of Long Beach High School in a few years ago, about the same time I was in high school back then. So I'll turn it over to Emil for an industry introduction for this session. Emil.
Yeah, so Wilson, Long Beach, Bruins, yeah. So I'm happy to be here to give you an introduction, and I have only a short time, so we'll fly like a drone quickly through the whole span of things and hopefully give you a feel for a little about what we've learned about drug development and rare diseases. And if there's a word to talk about drug development and rare diseases, it's resilience. Resilience is the word as a parent waking up each day and dealing with what happens in the day. It's what happens when you deal with the diagnosis itself. But it's what we deal with in drug development and rare because there's so much unknown, so much we have to figure out on the fly. And so we'll tell the story a little about who we are at Ultragenyx, but a little about my own career that leads us to where we are now and trying to develop drugs for many rare diseases. This is just my financial disclosure. I will touch on GTX-102. It is an investigational drug, and it's not approved by any regulatory authority. So Ultragenyx, you heard a little bit just a moment ago. 2010, me and my secretary were going to start a company. It always seems totally improbable. I did have a couple $1 million. My wife let me take that to do this, which was very generous of her. That was a big chunk of what we had, and the worry she had was like, that's probably all of it's going to go in the end. But you start with a vision to develop rare and ultra-rare disease treatments and to be very focused on products, getting to patients, doing it quickly. And in that time, we've now in 14 years, we're 1,300 employees globally. We've gotten 4 approved products. We're working on 20 different diseases. Angelman is one of them. We work on 20 different ones. Some are like Angelman, like CDKL5 deficiency, but there's a lot of others, all kinds of different treatments. And it's nothing more exciting, I can tell you, as a scientist and physician to be able to bring new treatments to patients. And we'll tell you a little about how that happens today. This is our mission, rare disease patients, and not only just getting drugs approved, but really it's about leading the future of rare disease medicine, which means change the methods, improve the process, the regulation, the design of things, make it more efficient, make it more possible to treat ultra-rare. And some of those issues will come up as I go through the talk. So just for anchoring, this is where I started, Harbor UCLA in Torrance. That was where my lab was, in that building. It was very dusty, it was old. But the truth was you could do good research there, and there's a lot of good research that went there. But there wasn't a lot of money, and working on genetic diseases and rare diseases, there wasn't really the kind of investment that goes on for a lot of other types of things. But the science was amazing, and we did good work there. And that was at very beginning. But as I started working on a disease called MPS1, a lysosomal storage disease, a terrible disease, it's my first connection to patients. And this is Mark Dant with his son Ryan. Ryan has MPS1. He's 5 years old. He got diagnosed at 3, and His father expected him to pass away by the time he was a teenager, and he was coming to me and say, "Can you save Ryan?" And he's handing me a check for $40,000. And that's not enough to do it, but there's a bond there. Scientists, we had a treatment idea that actually was working in dogs, and he says, "I'm going to devote everything we have to this." And that was my first connection to the personal, deep connection to the struggle, right in the battle. And he started raising more money, and we set off on a vision. We had no idea if we were going to get there, but I knew I couldn't not try, and we tried. Fortunately, a company brought in some money, and we were able to treat 10 patients. These are 10 kids, 9 kids with MPS I in this picture, and it was working. The treatment worked. What an amazing thing. But then the FDA said, well, that open label trial is not enough. You need a randomized trial now. It became a subject of a 60 Minutes episode, and then could we get enough money now all of a sudden? We'd already spent like $30 million. I'm thinking that's already an incredible amount of money when there's only 200 patients maybe we're going to treat in the US. And so we managed to figure out how to get the phase 3 done. And we got there, but what I began learning is that this process here, you can see the first few years on the top, getting to mark and then treating, and then that picture there with the company called BioMarin, those are all the people involved in the filing for approval. 150 people, almost as many people working on the drug as are going to get it treated. And then I learned how many technical skill sets and things you needed. It was just, and we spent $130 million or $150 million to get a treatment that was going to treat 200 people. That's kind of insane, right? But the truth is, what are you going to do? That's what it takes. And I've learned very early on that telling parents, being altruistic and saying, I really love to help your kid, but I don't have money to get the treatment developed. There's no benefit to that discussion, right? It's only failure. The truth is you got to get what you have to get it done. And it's hard to change the whole system, but that's what it is. So I wrote a book on this struggle. If you want to read about it, you can read about this book, Saving Ryan. But the most important part of the struggle is the fact that patients have a real big impact on developing drugs, just like your organization, ASF, will have such an impact. But the good part you see at the end of the book is that that's Ryan. He is now 35 and he's got married. I went to his wedding. You can get there. That's what we hope for everyone that has a rare disease, to get there. So this led me from academia to industry, because I learned something. It doesn't matter if I care about rare disease patients. If I don't have the money, I can't do anything. In order to get the money, you have to go to a for-profit company where then we can raise the money, and we have to deal with the money part. But if we get enough money, then we can treat more rare diseases and figure out how to make this work. And that's what I've been doing. I spent 11 years at BioMarin and now another 14 at Ultragenyx. But in my time, I've been involved in 11 drug approvals of all kinds of different diseases. And it can get done. And these, once approved, then kids with this disease, when they get diagnosed again, they're not told, no, there's no treatment. They said, no, there is a specific treatment. These are all specific treatments for those diseases. So what do we learn? Rare disease drug development is hard. As you probably know, small patient population, although Angelman has more than some of the others, I'll just be clear. One of the diseases we have has only 20 patients on the treatment in the US, but it worked and we got it approved. But all these diseases are complex and variable, and natural history has been collected, and many of you have participated, which is great. There's more natural history in Angelman than I've had in any program I've ever worked on, right? So that's good. You've done some good work laying the foundation. And the trials are expensive, and I'll talk a little about trial design and development. The problem is it's been getting harder and easier. That is, as there have been some rare disease successes, and the regulators and reimbursers and everyone says, oh good, it's doable. Now let's ask for this and ask for that and add more and more things, and then ultimately it gets more and more expensive. And I'm trying very hard to try to manage that, the time, how long it takes, and how long it costs. With brain disease and slower progressing disease, it can be more difficult because of this. But despite that, here's what it actually takes to file. Now I put this in, you might wonder, why am I showing you these numbers? I just want you to get a feel for what happens in the industry, because I didn't know it until I got there. So if we want to file a drug for approval with the FDA, what does that take? Well, it takes something like 100,000 to 300,000 pages of documents. All right, so it's on the order of, let's say, 1,000, 2,000 individual documents, 200, 300 page right? Some of the documents have 1 million hyperlinks between different pieces. It takes program software to connect all the documents. I just want you to understand the scale of this, right? It is mind-blowing. And in there are all these little bits of information that the FDA, someone FDA wants. That's what it takes. And it takes then a lot of people, and that's what's going on. And I can't change the system exactly, but we got to do it. This is what it is. It's not going to stop us, though. It just means it takes a bit of work. So the exciting thing is the technologies that are coming. They're actually things, and there's multiple technologies being applied to Angelman. That's what's exciting. Antisense oligonucleotides is one, and we're very excited about what we're doing in that, but there's so much more going on. And fortunately, getting Angelman syndrome is never what you hope for, but it does have some good features for being treatable. And I will say to you, there's a lot of diseases where the possibility of treatment is maybe none, because of what it happens to the kid that may be irreversible. But it's not a neurodegenerative disease. The biology is possible. You can make neurons talk to each other, but that's possible. That was figured out. And there's actually a fair number of Angelman patients. That means it's really more easy. It's not, when we did a 12-patient trial for that one disease, I had to move people from around the world to the United States for a year to run the trial. That's how hard it was to find patients. In this case, that's not true. And of course, a highly motivated patient community. So that keeps it going, and you guys have been amazing. So a lot of treatment strategies are working, including in some neuro diseases. But what I would say to you is the number of successes in neurologic diseases have been less. Neurology is hard. It's because your brain is complicated. Understanding and measuring it is harder, and therefore there have been some great successes, like a few here, but there's also been a lot of failed studies. So neurology makes it hard because it's so hard to measure and evaluate things. The brains are so complex. One of the challenges in all this is that there's a lot of variability. I'm sure you guys see all your other kids, you kind of know they have a lot of things in common, but a whole lot of weird different things and how they are. That variability becomes a challenge when you're trying to show if someone's getting better or not, and how do you measure it? And it's part of what we think a lot about. There's several areas I'm going to talk about now about drug design, which are important and what we do differently. One is about dosing, the other is about endpoints, and then we'll talk about trial design. Dosing, like how much drug do you give someone, becomes a very highly individual thing, because each kid's body is just different. And the drug may act the same way in all the different ones, but it's not the same degree. And so one of the things we like to do is use individualized titration strategy to try to get optimal, at least to get closer to optimal. And so finding the right dose with dynamic individualized titration, that's what we did in our early GTX 102 study. We switched to that strategy. We start you low, we titrate you up, and we look at how you're responding and try to make a sense, are we where we need to be or not? And from that, kind of learn what's the range of doses, what is the likely response. It helps us go a little faster, because otherwise if you set on a set dose, you may guess wrong and spend 2 years operating at the wrong dose. So creating this dynamic allows you to change and optimize on an individual case. And we also allow regimen optimization too. So this allows us some flexibility to more quickly get to an answer. This is what we did in the first dose titration trial that we did. I won't go through all the data, but we had 4 loading doses and then a maintenance doses. We started a group at low doses, titrated through to see where they would go, and then we started another cohort, but this time we started higher, because we sort of said, hey, you don't have to start as low, you can start higher. We start higher and move through. And what this does over time is it turns out all these patients are getting to about 12 to 14 mg doses. That's where it's coming out. There's some kids that respond to less. Not everyone is the same. This gives us an understanding of how much variation there is and gives us a better handle on how to think about dosing in the future. Now, one of the tricky areas, endpoints, how do you measure if you're better? Now, one of the weird things is the difference from you going to your doctor versus us doing an experiment. So you go into your doctor, and your doctor is evaluating your child with Angelman or something. You're not, and even for your own health, you don't go to the doctor and he doesn't measure one thing on you and tell you whether you're better or not, right? You don't have a single primary endpoint on your clinical care, right? Right? Who would decide everything on one? You'd ask a whole series of things and you pull it together and you come up with a feeling. How are you doing? How's your disease doing, right? But then we go into a trial and we say, no, we're going to do one primary endpoint. And we're going to decide everything on one thing. We're going to ignore everything else. That doesn't make a lot of sense, especially when you have a disease like Angelman, which everyone is different. Everyone has different things. I've been struggling with this from the beginning because the statisticians make us do one endpoint. The truth is all of us know there's more than one thing that matters. So why are we talking about one? And why are we just saying the one is the most important and none of the others? How do you rate them? Each person might rate a different one, right? It's the number one. So it's one of the real problems. And I've been struggling with this for a while. We've come up with, the guy working with me on the Ultragenyx program came up with this, what we call the multi-domain responder index. And I've been pitching it because I want you to understand it. The idea of this is another way to look at multi-domain, many different clinical problems a patient has, and look at them together and assess how you're doing. And I'm going to explain this to you because I think it's kind of important, and we are going to use it in our Angelman phase 3 so you can understand it. As I show you the data, I think one of the things I've heard from patients is when they look at the table, look at the data, they understand what's going on in the trial better than looking at a graph with a line on it. I think that's true. So I first developed this with MPS I. MPS I is a terrible disease. It has all kinds of physical problems, lung, heart, joints, every part of your body has a problem. And for those parents, the same heterogeneity, all kinds of different things going on. And what the idea of the multi-domain responder is to say, look, let's figure out what are the 4 to 6 clinical problems that are really important, the ones that really matter to patients, right, and parents. And let's just score each one. Do you get better by a lot? Or go worse by a lot? Or if you're kind of a little up and down, a little down, not big move, right? What you want to know is who gets better a lot. You want to figure out the big wins, right? Count those. If you're getting worse, that's certainly not good. So you count the big wins or big losses, and then you look across all 5 of those endpoints. It's kind of like a scorecard, right? You have 5 different endpoints. Did you win? How many did you win on? How many did you not win on? It's a pretty simple concept. So we did this for Ultragenyx. Now, this is a case we look at lung capacity, walking, sleeping, shoulder function, visual, eyesight, assess them throughout. What's a big win? What does that look like? What does a really big improvement look like? And then we score it. Now, this is data from a randomized trial. On the Ultragenyx treatment side are a list of patients, 24 through 45, and those are the 5 domains. The red boxes indicate they got better by a lot. And you can see on that side that the red is much more than yellow, and yellow is I got worse. So you can look and say, wow, the reds beat the yellows by a lot, right? On the left side was the kids who got placebo. There you can see that the yellows are more than the reds, right? And so you immediately get a sense that, for example, in visual acuity, the placebo patients, no change. But 5 of the patients had a substantial improvement in their vision, which was very important to them. It may have been a small fraction, but it was, for those kids, it was really important. Their visual acuity, which was poor, all 5 that had poor vision, those 5, got better. So you begin to learn more about what's going on in this approach. Now, we think it's a great way for analyzing Angelman syndrome and a more powerful method to capture a better feel for how kids are doing. So in Angelman, there's, of course, multiple domains. You know all the domains, and we've done analyses using this in Angelman syndrome as another way of looking at it. It doesn't mean the other ways are not useful. It's just another way of looking at the same data that helps you understand it. So when we did it with Angelman, this is very complex 2 slides. On the left side, cohort 4-7, those are the dose titration patients at day 338. So that's 1 year of treatment. As you can see there are 4 domains being shown. And you can see the result in the green. The green means they're better. And so you can see on the far right, all the patients but two. One had a decline in one domain, one had a 0, but everyone else had a usually 2 to 5 domains improvement, 2 to 4 domain improvement. On the right side is the most recent data, cohort A and B, but that's at 6 months, day 170. And there you can see all but 4 patients had at least one domain, usually 2 to 5 domain improvement, but you can see where they got better, and it gives you kind of a feel for how they're doing. The power of these methods is very high. These values change with just 12 or 20 patients. P-values are less than 0.01, right? They're very strong. This pattern of a change is a very strong statement toward the efficacy, and we feel very strong about this. And FDA has now accepted we can use this as a key secondary endpoint, which is important. It gives you another way to look at your data, and I think an important way of understanding what's happening on a patient-by-patient basis in terms of benefit. So that's one thing, and I want to move quickly along because I don't have a lot of time. So finally, proving a drug works. Last thing I want to talk about. The FDA loves randomized double-blind placebo-controlled trials. This is what they like because they feel this is the rigorous way to do it. And sometimes it's not possible. Natural history control trials, a lot of people want. I know in Angelman have asked us about doing it that way, where we just have some other patients we studied and we just compare how these guys are doing. The truth is it's hard to do this in Angelman that way because of a few things I'll touch on. But I just want to touch on these 2 ideas of a trial. Now, I've been involved in programs that did do a natural history control, but this is a disease called Batten's disease. These kids decline fast. Between age 2, they get diagnosed and they go into horrible situation within 2 years. Like they're very almost to death. So it's a very rapid thing. You can't do a randomized controlled trial. The control arm would be assigned, right, to decline and death. In this incredible situation, then natural history had been done historically, and it was so profound, the FDA allowed them to do the treated group and just compare. And you can see the treated group survive, the other patients are being lost. It's not hard to see the delta, it's big. So you can do this kind of study, but it has to be dramatic. It has to be like irrefutable score. These are very profound effects. When you talk about something like Angelman, it's going to be much harder to do that. Natural history is very flat. So you think that's pretty good. If it's flat, then you can be pretty confident if they're getting better, it's real. The problem is the endpoints. We ask cognition and Bailey. These are where we're asking questions, interpreting, can be biased. I believe the data we have are very solid and very real, but people ask the question. They still ask the question, is this placebo or not? Is it a real effect? The type of endpoints we have to do in a developmental disorder make it very hard because you can't be absolutely sure that the patient you study is like the natural history patient. Can we verify that? So it's very hard to get regulatory authorities to agree. Not only that, when we finally get approved now, it's not just the FDA, it's like all the reimbursers are saying, hey, I don't like the quality of your data. And they're looking for any excuse to say, hey, we don't think we want to pay for a treatment. I'll say to you, no matter what, at Ultragenyx, we get approved in the US, everything that get treated needs to get treated. We won't let the insurance company stop that. But I'm just saying to you, the better your data, the less trouble you have down the road, not just in the US, but everywhere. So we did meet with the FDA, and we've agreed on a randomized control design. At this point, they had suggested to us that we should do sham, not placebo. We're still doing the lumbar punctures. Sham means they don't actually give a saline injection to the control patient. They do the tap and get safety information for us, but they don't actually inject. I don't think it's truly a difference in safety. I think placebo would be better. However, the regulators feel that there's a question of ethical, and there was a sense that patients prefer sham. So we're doing the phase 3 sham. We don't want to delay and struggle through this discussion about how you do a control. It will be a randomized control. That means the control group will have procedures and tests that won't get treated, but when the 48 weeks is up, they will get treatment. So anyone in the trial will get access to treatment years before anyone else. So it's part of the contribution we need everyone to make in order to get this drug But we told the agency, and I'm telling you now, we're doing one control trial. And then the other types will do something else. But one control trial should be enough to prove that what you're seeing in these Angelman kids is real. All right? Prove it's real, but we don't have to keep doing that over and over again for multiple people. The endpoints of the Bayley-4 cognitive test. It's the best test. It basically, and I think, actually, I don't want to steal Kim's. Kim's going to go more in detail this, so I'll leave that to Kim to talk about. But these are the basic endpoints. She's going to go through the phase 3 trial. So right now, we're finalizing the protocol and planning the little pits addition. We're continuing to treat those phase 2 patients. We won't take people off the treatment. The key thing for some of you will be missense and other non-deletion type Angelman. We are going to do a study. We're planning the design right now. Our plan right now is that study is an open label study, not a randomized trial for those patients. So you've waited, but at least you won't be in the randomized trial. So there's a plus and upside to the wait. That trial will go on. The phase 3, we have to get started quickly. That trial will start soon after the phase 3 in 2025. And that will help get patients missense, UPD, and some of the other types, plus some age groups, like what about adults? We'll include adults. We're going to manage really young patients as well. We'll collect data in the hope, not proven yet, but the hope would be to use that data to help support that when the drug file goes in, that will have shown safety and efficacy in those studies to support the phase 3 data in the main population. That's our strategy. And we've done that strategy in multiple of the drugs before. So what do the Angelman community need for the next couple of years? Participate in trials. It feels like we have a lot of people want to be in the trial, so I don't know there's a problem, but it is definitely a burden. We know it. We're going to try to manage the testing burden as best we can. We are tuned to it. We're paying attention to it. I think it's also going to be important for you to provide the caregiver perspective on what's important for Angelman families. Because when we show results, someone's going to say, I don't care about that, but you might care a lot. They're going to hear what that is, right? They need to hear that my kid is sleeping through the night. Why is that a big deal versus not sleeping through the night? The people have told me, well, why does sleep matter? I said, oh, it matters when you're not getting it. So I don't know why I have to explain that to people, but apparently they have a problem with this. So you'll need to be active, and there may be questionnaires and things come out. I'm just saying to they come, please fill them and do them, because they need to hear your voice on why stuff matters and how much matters. And always keep us informed about your concerns. Don't hesitate. Send me an email or him or someone. Happy to hear everything. All right. So thank you for having me here. My time is probably a little bit long. Kim speaks fast, but thank you.